From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 1, recorded on September 27th, 2019. I'm Vincent Racaniello, and this is a brand new podcast all about the brain. The goal here is to have some academic scientists talk all about neuroscience, and this is the brainchild of one of the co-hosts. We'll introduce those to you in a moment, but he's sitting with me here in New York City. He is an MD PhD student at Columbia University Vagelos College of Physicians and Surgeons, Ori Lieberman.
Hi, Vincent.
I shouldn't say welcome because this is your idea.
Well, yes, welcome to you all. Yeah.
And also joining us from Nashville, Tennessee, Erin Calipari. Welcome to TWiN.
Thank you. Hi.
And from Salt Lake City, it's not Nell Zeldy. Jason Shepard. Welcome to TWiN.
Hi, Vincent. Hi, Ori. Yeah, well, looking forward to this.
And Ori, it's great that you were able to gather this terrific group.
There were a lot of rejections on the way. So,
so what are you trying to say?
You guys are who I could scrape up.
These are
Thanks.
the individuals with foresight and prescience and who want to communicate science to the public, right? It's really important. So
Exactly.
we have another host joining us in the future, right?
Yeah, so Andres Bendeski here will join intermittently, I think.
Okay,
Yeah.
and he's here at Columbia as well.
Yes.
So here in New York, it's a gorgeous fall day. It's just a few clouds in the sky. It's like in the 70s.
We should record outside.
Oh, yeah. Where was I recently? They wanted to record outside, but it's a bit noisy. There's no power. What's the weather in Nashville? Is it beautiful?
Oh, it's hot. No, it's so hot. I moved down here from New York City, and I'm just like, I was like, oh, it's not going to be as cold in the winter, and that's true. But there's a couple-week period right now where it's like unbearably hot. So it's humid and hot, and I don't do outside that much this time of year.
How about Salt Lake? What's it like there?
It's perfect. This is my favorite time of the year here. It's not too hot, not too cold. Although actually this weekend we may get our first snowfall in the mountains, or significant snowfall. So the skiers are excited.
Nice. I was out in Reno this week, and they already had snow in early September.
Guess global warming is not working. I don't know.
Well, I don't think there's a lot of snow, but
Yeah.
it was something there. So this, as we said, this is a brand new podcast. The focus will be neuroscience, and it was Ori's idea. He came and talked to me, and I said, well, if you take care of the guests and the topics, I'm happy to help out. So I'll be an observer, but on this today, we're going to learn all about these co-hosts and what they've done and what they're doing and so forth. Before we start that, if you like TWiN, and I know it's early, but we do other podcasts over at microbe.tv. If you like them, consider supporting us financially. You can go to microbe.tv/contribute for ways that you can do that, and we'll talk more about that later. Okay, so let's have each of us talk a little bit about our training and what we do. Of course, I'm the odd person out because I do viruses. I actually destroy the central nervous system, where you try and understand how it's developing and working. So that's my connection. I work on neurotropic viruses.
Yeah.
So, Erin, let's start with you. Where are you from originally?
Oh, yeah. I always ask other people where they're from and then try to say something relatable about that area. I've moved I think 10, 12 times in my life. So I've kind of a little bit of everything up and down the East Coast to the middle. I did my undergraduate, though, at the University of Massachusetts in Amherst. And so Massachusetts I know pretty well. I started studying the neurotoxic effects of MDMA or ecstasy on the serotonin system. And so I was really excited in undergrad about kind of, neural control of behavior and how drugs affected brain function. I ended up moving from there to Winston-Salem, North Carolina. And so I did my PhD at Wake Forest School of Medicine, where I then kind of became an analytical chemist. I was doing a lot of sub-second monitoring of neurotransmitter species, mostly dopamine and other catecholamines, and trying to understand how abused drugs like cocaine and amphetamine and even kind of prescription abused drugs like Ritalin affected dopamine function to drive behaviors that are characteristic of substance use disorder. Then I moved from there to New York City, and I was at Icahn School of Medicine at Mount Sinai in a lab that looked at the genetics of substance use disorder. So we were doing a lot of big data, next-gen RNA sequencing. I was doing a lot of circuit control of behavior work. And then 2 years ago, I started my own lab at Vanderbilt University in Nashville, Tennessee, where I look at neural circuit control of decision making and how interactions between epigenetic factors and activity dependent gene regulation control how animals make decisions, and then how substance use disorder, depression, anxiety dysregulate this to drive kind of these phenotypic characteristics of these disorders. So my lab is a little bit broad, some big data, some optical tools, some sequencing approaches, but we try to kind of focus on big picture questions and then integrate new technology to kind of answer those in fun and interesting ways. And so, I've kind of had quite the path, but all neuroscience has really been my background, neuroscience and a little bit of endocrinology here and there.
So I also was at Sinai. I got my PhD there.
Oh, wow.
But much earlier than you were there.
You said some time ago. Yes, Sinai is great. It's a funny thing because it's on that like little corner of Central Park. So it's a kind of beautiful thing. You take a few steps over, have lunch in the park, come back to the hospital and work. And so, yeah, I really enjoyed being up there, although it's a little bit cheaper down here in Nashville. So I don't, I'm not as sad when I go home to my apartment that actually, can fit some furniture and maybe a few people instead of just one. But
who did you work with at Sinai? What was the name?
I was with Eric Nestler.
Okay.
So, yeah.
So I remember the park because when centrifuge rotors would get derated in the old days, before titanium, they were aluminum, and you could only go so many revolutions, and then they would say you can't use it anymore. We used to take those and go to Central Park and chuck them. Have rotor chucking contests.
That's pretty great. I mean, that's not surprising. I've seen a lot of things in Central Park that would not actually make the top of my list for the weirdest. So
it would make big divots in the grass. Right? It was like throwing a shot put. Of course, that's not the only thing I remember. But so you don't have any, as far as I can tell, you don't have a detectable accent. Is that fair?
Yeah, so my mom is Midwestern, so that's kind of the every person's accent. So she's from Missouri. My dad's from Pittsburgh, Pennsylvania. So he actually does have an accent, and it's not the best. So I think we've kind of defaulted to the kind of middle ground. Although down here, people think I sound like a New Yorker. So they always ask me where I'm from. And I say, I'm from Nashville. And they say, no, but where are you really from? Mm-hmm. Mm-hmm. And so that's usually what they guess. But I think it's just because it's more of a Northeastern accent maybe than a Southern one.
So your lab is a couple of years old, you said?
Yes. 2 in November.
Wow. That's pretty exciting. So I bet it's full of students already, right?
Yeah, so I have 5 postdocs and 3 PhD students and some undergrads and a master's student. So we're quite large, actually.
It's a lot of people.
Yeah, no, it's funny. I got really, really good people. And so then there was space for more people, and they're a really great collaborative group. And I've been so excited to see how they kind of work together and value collaboration over kind of their own personal success. And so as a result, they've all done better. And it's a really nice place to work with, nice people who care about science. It's awesome. I am very, very happy.
So you're in the Department of Pharmacology. That must be in the medical school, right?
So we have a weird system. I'm in the Department of Pharmacology primarily. And so it's actually part of the basic sciences, which goes through the undergrad institution. But my space is in the medical school. And I also have an affiliation with psychiatry and with the physiology department here. So we're kind of this weird intermediate, but it's actually really awesome because our academic mission is driven by the undergrad, which is more of a discovery-based mission than a kind of hospital-based one. But then we have a lot of resources that are associated with kind of having really strong clinical populations. And, there's a lot of interaction interaction with clinicians. So it's actually a really unique place because I've always been at only medical schools for my career. And I really, really like that structure because they value the kind of discovery part of it, but also the translation. And so it's a pretty cool environment.
Are you teaching at all there?
I do because I like teaching, but it's not required of me. So I teach some graduate courses. Every once in a while, I'll do an undergraduate, and by courses, I mean like lectures. I don't do like full course stuff. Our structure is there's not as much teaching, but that's kind of good. You can kind of pick it up where you want to. So I do some teaching and speaking and forums and panels and things because I think it's important, but it's not required. So, it depends who you talk to here, how much teaching people do.
Yeah, I know it. I'm at a medical school and never was required to do any teaching, but I liked it, so I did it.
And you taught this morning? Did you teach this morning? So
this morning I taught to medical students, which is unusual because I haven't taught them in many years. They broke up the first year class into 4 or 5 and had some basic scientists talk about what they do because otherwise the medical students never hear any of that, right? So I talked about viruses that cause paralysis, polio, and then the one that's doing it now, Entero 68. And they're interested because it's real life application of what they're learning. I used to teach medical students a lot, but over the years they kind of reduced it, and now mostly it's the clinicians who teach them basic science, which I think is a bit of a problem, right? But they decide
when last did they do basic science?
But that's because I always would get this question, is it going to be on the boards, right?
Exactly.
But actually there is basic virology on the board.
There are, yeah.
And so I'm sure there's neuroscience as well, right? Did you take the boards yet, or?
Yeah, I took there's 3 boards in medical. I took the first one before I went to lab.
And so there was virology on it.
There was virology. And I think you taught us 2 lectures, maybe, all of virology in 2 hours.
I used to do 7, and it was then 2, and now it's 0. Although now the course director says she would like to get me back. So I said, you know where I am, right? So
I'd always get frustrated by that question about, is it going to be in the test? And I would say, well, is that what you're going to say to your patient when you don't know something? Oh, it wasn't in the test.
That is exactly what I tell them. I said, someday a patient's going to ask you something and you're not going to know, and they're going to think you're a jerk because you don't know. And so you need to learn this. And I started teaching an undergraduate virology course 10 years ago because I like teaching, and that's a blast. I kind of wish we had undergrads here at the medical center. Yeah. Because, 1 mile on the subway is enough to prevent them from coming up here pretty much.
Yeah, I was saying that was a thing at Sinai as well. There were just no undergrads because it's, they could just go to any of the schools they actually go to. And so Vanderbilt has tons of undergrads that are here. And so it's actually really nice to have them involved in the kinds of research things you do. And it's very, very different than places like Columbia or Sinai where they were just not
separate.
there.
Yeah. Yeah. I like when they're together. Because at Sinai, I remember as a grad student, I took courses with medical students pretty much. Microbiology, biochemistry, they're the medical student courses, and they're not focused for graduate students, right? It's a different focus. Jason, where are you from?
Yeah, so my story is also somewhat convoluted. So I was actually born in South Africa, and my family is from South Africa. But then we moved to New Zealand when I was 14. So that's why my accent is so weird. Although now of course my friends in New Zealand tell me that I have an American accent, which is unfortunate. But so when I was 14, of course, it was kind of interesting because my brother was 3 years younger, and he picked up a full New Zealand accent, but my accent's always been this sort of mixed up thing. So that just shows you the power of the plasticity of the brain, and we can talk about that at some point. But it's also something that I've been interested in figuring out for my own research. So in New Zealand, I went to high school and then went to undergrad. I went to undergrad at the University of Otago, which is at the bottom of the South Island of New Zealand. So basically nothing between Antarctica and that city. But it was a great place, beautiful place to do undergrad. It was a college town, and a bit raucous, but a lot of fun. So really a great place to do your undergrad. But I was always interested in being a scientist. So I did actually the first year. So in New Zealand, there's 2 medical schools, and basically you have to decide right at the end of high school if you want to do medicine. And so I started doing medicine at the University of Otago, and it's basically this 1-year concentrated pre-med. Everyone who wants to do medicine has to do this concentrated pre-med year, and then they choose the top 100 scores or grades at the end for the people who could actually continue to do medicine. And we had one elective, so the pre-med was the usual physics, chemistry, biology, and you could choose one elective, and that elective I chose was neuropsychology. And after that elective, I was like, okay, I think I just want to learn how the brain works, and so I decided to skip out of medicine and do an undergrad major in neuroscience. And haven't looked back since. So that was my fork in the road, whether I do medicine or not, but decided to do research. And my 4th year, so the New Zealand undergrad system's like the British. So you do a 3-year bachelor's, and if you want to do a PhD, you have to do an extra year called an honors degree. And that year you can do a thesis. But I did something unusual in that I decided to do that thesis as an exchange student at UC Irvine. So I went to UC Irvine. That was my first time going to America, and landed in Orange County. That was, I would say it's a very formative experience of sort of cultural differences, but ended up working with Frank Lafurla on an Alzheimer's mouse model. But that exchange year, so everything I did there was I got credit for back home, but I was able to then apply for grad school while I was there, and of course much easier to interview someone who's in America rather than have to fly them out from New Zealand. So that's really how I ended up being able to go to grad school in the States. Did my PhD then at Johns Hopkins University with Paul Worley and Rick Huganier in the Department of Neuroscience at Hopkins, which is an amazing place. Absolutely loved my PhD experience. And then moved to MIT for my postdoc with Mark Baer, and swapped Baltimore for Boston, which was an upgrade. Love Boston.
I've been to both those places and lived in Boston, and I would agree.
I'm from Baltimore, so I'll take offense to that.
No, Baltimore has its charm, as they say, but
Charm City, yeah.
Charm City. Well, it was funny because when I first moved to Baltimore, I was sort of renting an apartment right close to Mount Vernon, which is this really nice area. But they started filming The Wire, the series, right outside my apartment. And at the time, I had no idea what this was. And when it came out, I started watching the first few episodes. I actually recognized my apartment, and I had to tell my mom not to watch The Wire because I think she would have completely freaked out. I was like, "What is where's he living?"
That's funny. Is it a good show? I've never watched it.
It was good.
Yeah,
it's a brilliant show. I thought it was. Yeah,
it's a great show. It's so good.
It's really good. So then after MIT, moved to University of Utah. So that was almost 7 years ago, which is crazy to think about. But and started my lab at the University of Utah in the School of Medicine in the neurobiology and anatomy department.
You've moved around also.
Moved around.
Wow, that's quite a bit. I think landing in Orange County as your first U.S. experience is not bad. It's pretty nice there.
It's nice, but I can tell you. But there was a couple of issues. One is I didn't have any money, and I didn't have a car. And then I was stuck in the undergrad dorms, but I was doing graduate level research and classes. So socially, I was sort of this no man's land, and no one really knew who I was. And then they were like, "Who's this guy that's in these PhD classes? He's got a funny accent, and he's not shy." And so they didn't really know what to do with me, but it paid off in the end. It was definitely a great move.
Not having a car in California is a big issue, right?
Exactly. Yeah.
But the weather is great, right?
Weather was great. Yeah, and I couldn't complain about that for sure.
So what is your research interest nowadays?
Yeah, so my sort of the main interest that I've had throughout my career is trying to understand how the brain stores information for long periods of time, but really trying to figure out how that works at the molecular level. So genes and proteins, how do they transduce experiences into changes in the brain, and particular looking at synaptic changes. So how do the synapses between neurons store information? And really taken a very focused approach by working on one particular gene called ARC, A-R-C. And the funny thing is that every time I think that we've solved what that one gene does, it throws us surprises, and then I can't let go of it. But we know that this gene is absolutely essential for long-term memory consolidation. So mice where we've deleted the gene, they basically can't remember any long-term information. So they can actually learn. So the learning part of making the memory, encoding the memory is intact. But this gene seems to be really essential for turning the learning into long-term information and storage in the brain. So we've been trying to figure out why that's the case. Why is this one gene so essential? And that has included looking at in vivo, so looking at how this gene regulates synapses and circuits in vivo and in the intact mice, all the way to trying to figure out what the protein itself, so what is the biochemistry of the protein? And then a few years ago, while we were trying to figure out the biochemistry, we made a really odd observation. So we started a collaboration with Adam Frost's group to look at look at the actual protein itself, the structure of the protein, by purifying it. And when we did that, we actually found that the protein, the purified protein, spontaneously formed these large oligomeric structures that when you look at them under EM, look like soccer balls. So of course you know what these structures were, but when I first looked at them, I was like, what are these? This is a neuronal gene that's making some sort of weird soccer ball structure. What could they be? And of course, it ended up finding out that they look like these viral capsids that HIV makes. So that one observation has taken us down a really fun rabbit hole, and it turns out that ARC itself is derived from an ancient retrotransposon or retro element that contains the SCAG domain that forms the backbone of the capsid in all retroviruses, including HIV. So we think we've stumbled on this new kind of biology where this gene that perhaps accidentally got inserted, the sequences got inserted into an ancient ancestor, became useful for evolution to turn into a gene that somehow is important for memory.
And we did that paper on TWiV. It was in January 2018, number 477, and the episode was called Raiders of the Lost Ark.
That's great.
And I hope we have some cool titles also on this podcast.
He's warning me, yeah.
It's hard because I had a lot of training from Alan Dove, one of our TWiV co-hosts. Do humans have an ARC gene, Jason?
They do. So it's extremely. So what's interesting is extremely conserved in tetrapods, so land animals. So everything from birds to humans. So actually, at the amino acid level, between mice and humans, it's conserved it's over 90% conserved. So it's highly conserved in that lineage. But then there's no fish gene. And most invertebrates don't seem to have this gene either. So we're trying to figure out whether there's other genes that perform similar functions, or does ARC confer something really unique that land animals needed to adapt to all the environments that you have on land.
So in humans, are there any memory defects that map to the ARC gene?
Yeah, so it's interesting. We don't really know of particular mutations or SNPs in the gene itself, but there's a lot of mutations and SNPs in genes that we know regulate the expression of ARC. And in fact, most of those are detrimental. So there's intellectual disability, autism spectrum disorders, schizophrenia. A lot of these neurodevelopmental disorders seem to converge on the regulation of just a few genes. But it seems like if you mutate ARC itself, that maybe it's too detrimental. I mean, that's sort of hand-waving, but it's curious that a lot of the pathways that feed into expression of ARC are disrupted, but the gene itself is not.
Very cool. So neuroscience is broad. It's great. And speaking of memory, maybe someday we can get Eric Kandel to sit next to you, Ori, and we can do a twin with him.
Yeah, that'd be great.
I bet Jason would like that too, right?
For sure. No, I'd love that.
Ori, tell us about yourself.
So I was born in Chicago and then moved to Baltimore. Spent most of my life there. In the suburbs outside of Baltimore. And then
the suburbs.
Suburbs, yeah. Could have been anywhere in the U.S., yeah. And then went to University of Maryland, College Park.
Okay.
And I actually, in high school, I spent a summer in a lab at Hopkins in Jay Baraban's lab. So I remember seeing Worley and Huguenier written on equipment and stuff like that. And then at Maryland, kind of got into microbiology research and realized that this is what I wanted to do. So came here and joined the MD PhD program and had a field switch, I guess, to neuroscience at some point. And now I'm here.
Why do you think you had a field switch?
Neuroscience is very strong here. There's a lot of options, I think. So, and I was also primarily looking for a mentor who could kind of give me freedom, but also guidance in kind of a weird hybrid. And my PI in the lab that I'm in now fit that. So I was a little bit agnostic to topic.
Who's your PI?
David Sulzer.
And what do you work on?
David Sulzer does such cool stuff. Also, he's tell him what you work on first, but then we should talk about the fact that he teaches animals to play instruments.
Yes, he is an enigma in some ways. So I originally joined his lab to study Parkinson's disease, which he is kind of, I would say, half the lab works on. And then by accident got into developmental questions. So I was trying to learn how to patch clamp, which I'm sure we'll talk about at some point. And then it's much easier to patch from young mice, and realized that young mice are firing action potentials and have different synaptic properties than older mice, and have essentially focused for the last 4 years on how these changes happen during development. What are the molecular pathways, circuit pathways, and stuff like that.
So are you finished with so you're an MD PhD student, right?
Yes.
Are you almost done with your PhD part?
So I'm defending November 20th. So.
The same day we have a twin, right?
And do it after. So I should be writing, yeah.
And then what do you do after that?
So then I go back to med school in January.
Okay.
And I have a year worth of rotations left to finish.
And then after that, what happens?
I have no idea. I'll either do residency or do a postdoc, but it kind of switches back and forth. Yeah.
Go ahead.
I'm always curious about MD PhD students because I feel like not that many end up doing both. They at some point they decide that they either love the research or they love the seeing patients, but not both. So it sounds like you're actually you're torn between the two. Yeah,
so I kind of am. The MD was, so I love being at the bench and I love the research. The MD was a little bit of an accident, I think. And so I'm trying to figure out the right way to balance the two and maybe have some clinical exposure, but mostly focus on the research.
I see.
Well, the fact that you wanted to start a podcast tells me that you're not going to be a doc. Most likely.
Okay.
Because they're very focused and, don't want to spend time doing things like this. But we'll see. So you may leave at some point, right? In fact, you probably will.
Yeah, hopefully.
Because you're not going to stay here forever unless you end up doing something here, right?
So I'll be here for another year at Columbia for a year and a half more. Okay. And then we'll see where it takes me.
And it's okay because we have Skype and we could bring you in. And I assume you want to stay with TWiN as long as you can, right?
As long as you guys will do it. Yeah.
Well, we can. I'm sure people will get really excited about it and I mean, I love doing podcasts. So let me tell you a little bit about me, although I suspect the initial listeners of TWiN are going to be people coming from all the other podcasts that we do. So they know me. But for new people that are coming, I grew up in New Jersey, which I can see out my window here, which you can see.
It's still there, yeah.
And my father was a doctor. My mother was an English teacher. And that is my formation, basically. I was interested in science. I used to think that medicine was science, right? But then I ended up going into science, and I can write because my mother was an English teacher, I guess.
Same here.
Kind of stumbled into a PhD program. I liked science, but graduated from Cornell with a biology major, but I didn't know what I wanted to do. My dad wanted me to go to medical school, but I didn't want to, and I didn't develop any alternatives. So I worked as a microbiology lab tech for a year, and I got hooked on microbiology and viruses, and got a PhD with Peter Palese at Mount Sinai, working on influenza viruses. Then I did a postdoc with David Baltimore. So that would be 1979-ish. He had just got the Nobel Prize for discovering reverse transcriptase. His lab was really an interesting place.
Oh, yeah.
And I worked on poliovirus. And then came here in 1982 with poliovirus, started my lab, and I've been working since then on polio and related neurotropic viruses. And about, 15 years ago, I got involved in writing a virology textbook. And then that started all this communication stuff, because then I said, well, now I know about viruses, not just poliovirus. So I started blogging. I started teaching an undergraduate virology course, which I still do to this day. I started podcasting just because I thought it would be pretty cool to have a This Week in Virology. Who would listen? And little did I know that a lot of people did want to listen. And so we have This Week in Virology, This Week in Microbiology, This Week in Parasitism, This Week in Evolution with Nell Zeldy, who's out there with Jason in Salt Lake City, and Immune. And they're all people who like to hear scientists talk to one another. And I'm really lucky to have collected scientists from different fields to do that. And they're willing to spend the time, and the listeners love it. We have many, many thousands of listeners. We have made over 10 million total episode downloads. We over 1,000 episodes of all the different podcasts downloaded over 10 million times. We have thousands of listeners, depending on the podcast. We have many thousands or tens of thousands. And there are a lot of people out there who want to learn science and hear the scientists talk about it. And that's why I'm doing this. And in the meantime, my lab has gotten smaller and smaller because I think this is more important in a way. Although being at a medical school, I have to have a lab. Otherwise, I should leave. Right. So I'm trying to juggle that, and I keep a small lab. But I love teaching people. And now we get asked to do podcasts all over the place. I was in Reno last week. I was in Denmark the week before. In August, I was in Sweden. I'm going to Galveston next week. So it goes on and on. This is all for podcasting, not for seminars. That ended a while ago. But and everywhere I go, students and postdocs want to talk about communicating science, and that's something that we should be teaching our trainees, I think. So
I completely agree. There's no training in any of our, training lineage of how to communicate science to non-scientists. And it's such an important part of kind of how you explain why science is important, especially basic science that may not directly relate to the discovery of a medication or something. People can relate to that, but how, why we do this and why it's important.
I love pointing out how things that are therapeutically useful started as, let's just look and see what's going on, right? I mean, there's so many examples of that.
Yeah. No, but also I think the excitement about science, that's something that is kind of missed when we write a paper and it's dry and something like that. But it's exciting. I mean, every day you're going in and not every day, but you're finding things that are new, right? That no one else knows.
Every few years, right?
Yeah.
Someone said once that finding something out is amazing because no one else knows at that moment, right?
Yeah.
But people say they love hearing us talk about science, how it gets done. They like hearing that we're normal people like them, you know.
Yeah, exactly.
Semi-normal.
Putting a human face.
Put a human face on science, totally.
Yep.
And they particularly like us talking about how the paper is a linear progression of the story, but it doesn't actually happen that way, right? So that's what we're doing. And so we have, 3 people on this podcast that have different interests in neuroscience. So hopefully we'll be exploring that. And so that leads me to ask you, Ori, you had the idea to do this. I mean, I'd always thought about it, but I'm not a neuroscientist. So, but you came and you said you wanted to do this. So what are your goals for TWiN?
So I think there are 2 goals. One is to kind of spread information about neuroscience. And the second, and in some ways more important, is to spread the idea of understanding how neuroscience is done and how we understand the things that we know now about the brain, and where that can go from here. So I think that those 2 will be accomplished on this with the 4 of us, hopefully.
So one of the ways we'll do that is by talking about papers, right?
Right. So I think that for the most part, we'll do one paper every month, every episode. Mm-hmm. And we can kind of talk about what led up to the paper, what the authors did in the paper, and what it means for going forward for the field.
And because we have 3 or 4 different areas of interest, presumably people will pick something in their field and we'll get to experience, addiction or memory or patch clamping.
Yeah, and I think we can bring other people in as if we can bring in an author of the paper or someone who's visiting Columbia or Utah or Vanderbilt, can kind of bring in other expertise.
Yeah, that's something we can do for sure. Because each of us have a seminar program or multiple seminar programs. People come through, and so we should keep our eyes out and say, "Hey, so and so is coming," because we can always connect.
No, I like that idea of getting some guests on here as well. I think the first couple we just choose papers that will introduce some really basic topics about brain function, and then move on from there.
Yeah, and then so will you guys be at SFN in a couple weeks?
Yeah, it sounds great. I'm unfortunately not going to be there this year. Yeah, but I am actually going to be in Columbia in January, so I'm giving a seminar, I think, on the 7th of January, something like that.
Oh, okay. We can do a TWiN here together, yeah.
It'll be fun.
Yeah, if any of you come to New York, for sure, let us know and we can whip one up. But the idea is to do this monthly, right?
Yeah, monthly. And even though it's This Week in Neuroscience, but...
Well, yeah, we'll talk about what's happening on a weekly basis every month, right?
Yeah.
But many of the other podcasts I do are monthly or twice a month, but we still call them This Week. I don't know, just following the brand, I guess, right?
Yeah.
So that's cool. And there are other areas of neuroscience that are not represented here, right? Lots.
Many.
Oh, yeah.
Yeah. So if we could get if anyone is listening and wants to join and kind of bring in other parts of neuroscience, that would also be great.
Yeah, even if it's one-off, come and talk about worms or flies and aplesia, right?
Yeah, we can bring Erin in. Yeah.
All right, that would be great. So that's This Week in Neuroscience, and as I said before, we're lucky that we have people who are willing to do this. So it's greatly appreciated. Anything else, Ori, before we sign off?
No, maybe. Well, we could do picks next time.
Yeah,
maybe. I didn't prepare one, so you.
Yeah, so over on TWiV and which one? Any other? Immune, we do picks of the week where we share some sciencey thing that we like, and we could do that here. Probably lots of neuroscience related things that we could pull in, right?
Yeah.
Yeah.
Okay, so that.
Well, yeah. Thanks for the opportunity, Oriel. I think this is going to be fun.
Yeah. I'm gonna learn a lot. Let's put it that way. I'm looking
I
forward.
probably will too, and this is my field. So
Yes.
So
me
I
too,
guess
yeah.
the bottom line here is that neuroscience is so broad and everyone focuses. So Erin, you probably don't read much about memory, right?
I do a good amount of memory, but my background's psychology. So,
Okay.
but yeah, that's the thing, right? It's like, you have people like me who do computational models of, learning, and I pull in psychology, but maybe I don't know as much about, cell biology as I should.
Yeah.
Yeah, but this is what I love about neuroscience is that we're all trying to figure out how the brain works, but there's all different kinds of analysis, and I think the real challenge in the end is to synthesize them, everything from behavior to molecules, and I think that is the challenge that neuroscience faces that a lot of other fields don't have, but I think that getting people together like this that have complementary but different views on things or different experiences, that's key. And so, for example, like Erin does some work on activity dependent genes, and that's sort of the bread and butter of my lab. So there's always something that we could relate to, but I think it's what's fun is having these different viewpoints.
For sure. And we should probably do papers from time to time from our hosts' laboratories, right? Just to kind of flush out what it is that we are doing.
Yeah,
right?
that'd be
So
great.
Yeah.
you and Jason
definitely.
and Erin, that would be fun. So, but the plan is next few episodes to do some basics, Ori, is that right?
Yeah, so I think we'll pick some classic papers and that will kind of go from there to principles in neuroscience that, have changed the way the field has gone.
Great.
Yeah.
Looking forward to that. Yep. All right. So TWiN will be, by the time you hear this, it'll be available wherever fine podcasts are distributed, like Apple Podcasts. And, you'll probably listen on your phone or tablet using a podcast app. You can just search for TWiN or This Week in Neuroscience and subscribe. That would really help us to build up the number of listeners. You can go to microbe.tv/TWiN for show notes and links to papers that we talk about, and for downloading as well. And if you have questions, we'd love to get them. I can't answer them, but everyone else on the show will. TWiN@microbe.tv. And if you really like what we do, all of us do this on our own time. We have very little support for it. And so we depend on our listeners, and we have several ways that you can contribute. Go to microbe.tv/contribute. You could use, say, PayPal or Patreon and give us $1 a month. And you can also buy merchandise. We'll make some TWiN merchandise. We have a cool logo, right?
Yeah. I tweeted it out today.
Yeah, which we had made. So check it out, microbe.tv/contribute. I have to say, this is the first buzzing I've ever had on 1016 podcasts. Weird. Must be the nerves.
Yeah.
TWiN so far is Ori Lieberman. You can find him on Twitter, Ori Lieberman. Thanks, Ori.
Yeah, thank you for having us.
Thanks for having the idea. And how did you know to come here to start a podcast?
I've been listening to TWiV for a while.
Okay.
So, yeah.
Cool. Erin Calipari's at the Erin Calipari over on Twitter. Thanks for joining us, Erin.
Yeah, thank you.
Jason Shepard on Twitter is Jason Synaptic. That's cool. Thanks, Jason.
Yeah, thanks, Vincent. No, I've also been a fan of your podcast for a long time. So, of course, also a fan of Nell's. So happy to contribute to this.
Very cool. Well, the more podcasts we do, the more everyone's going to know everyone else, right? That's pretty cool. I'm Vincent Racaniello. You can find me at virology.ws. And we have no one to thank yet, but hopefully we will at some point. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll see you next time.